All right. Hi, Danny.
Hey there, Belinda. How you doing?
I'm
Can you
all
hear me?
right, sir. How are you?
I'm doing really well. Yeah.
How was the
Um,
stream today?
really good. Yeah, we kind of went all over the place today, but it was some good stuff. And right before we raided in today, I showed that video about with Richard Feynman, the physicist, and talking about kind of the joy of like sharing science with fellow scientists and other people who are interested in stuff too. I sent you this clip a while ago, Belinda. I don't know if you ever get a chance to look at it, but
Yeah, yeah, yeah.
it reminds me of our crossovers every Wednesday, and yeah, so I was eager to share that with the chat. And hello, Moon Pie. We're getting some cat activity here too. Wow, everything's happening at once. Come on up, Moon Pie. Come here. Yeah,
Actually,
I've also
so
got some lasagna that Lordy made on her stream today too. So,
yeah, she was
and
doing
then
the charity event where she was making the
yeah, she raised
lasagnas.
like $420 for that or something like that.
Yeah. Oh,
Pretty
she was
incredible.
she was crushing it today. I mean, she always does, but it was
Yeah.
really really cool to see like how much was raised, and then like all the lasagnas too like looked really good, and she puts garlic in there too. I'm like, oh yeah.
But I've never had lasagna as good as what Lordy makes. I'm not always the biggest fan of lasagna. You know, apologies to Garfield and Co. But Lordy makes the most incredible lasagnas. They are delightful. Like there's a wonderful kind of like citrusy background note to them
Ooh.
because she puts lemon and lime in them. I think,
I,
and
I
they're
it's like the brilliance of someone who knows how to cook, where it's a vegetarian one too, where it's not just red sauce
yeah.
and some cheese, right? But you actually have
Yep.
thought about the flavors and how. Yeah, guys, go
And
follow
there's
Lordy.
all of these different notes that are all playing simultaneously in it. It's like a symphony. It's kind of amazing.
I was telling Lordy
Yeah.
today, I'm like, I can't wait until the day I get to have one of her dishes because they always look
I
so
know, right?
good. Yeah.
I am incredibly lucky to be able to live in the same house as Lordy and eat some of her cooking. It's incredible stuff. It really is.
And then yeah, that clip actually because I'm everywhere at once. I always have
Uh
you,
huh.
if you're live on my laptop next to me, I have you pulled up, and I actually saw you playing that clip of the Feynman clip. And I was like,
Yeah,
oh, I
yeah.
think he's playing it. That's cool. That's also how I saw
Nothing gets
you.
past you, Balloon.
You were playing the
Yeah.
clip of when you were on that news station too, with the. It was like that
Yeah, yeah.
other. Was it another paleontologist as well who was on that?
Yeah, yeah. From the UK. Yep.
Yeah, and
Um,
it had like the old office background and everything. It was like, oh yeah,
yep.
that looks so cool. To like something we see on Twitch, but then on like TV.
Yeah, yeah. It was that interview went a lot better than I was thinking it would go, and grateful for that. So it's really nice to have that clip and be able to show that whenever we're talking about like commercial paleontology and that kind of thing. Um,
Yeah, about
yeah,
like the different lands too, and like the privatization of lands and like hiring out. It's a complicated. Well, I want to say mess, but I feel like it's.
mess is fair. That's very fair. Yeah, um, yeah, yeah. But anyway, yeah, and yeah, Nicolas Cage buying stolen fossils. There you go, Timmer's Crutch. That is very much a thing that happened. So
Was it The
buyers
Rock?
beware. Do not buy dinosaur fossils on the open market like that. You might end up having to forfeit them to the Mongolian government when it turns out they were smuggled out of there.
Was
Yeah.
it Danny The Rock who made a replica?
Oh, he purchased a replica. Yeah, yeah.
Yeah.
Dwayne the Rock Johnson purchased a replica of Stan, the T Rex, and good on him for doing that. You know, a lot of people were afraid that he had purchased the original because the original kind of went missing and nobody knew who bought it. And then this shows up in the background of some like Zoom interview that he did at one point, and it turns out no, that was just a replica. That was not the original, and so it was very much ethical and on the up and up. So good on
Yeah,
him. Dwayne
I
The
I,
Rock
I
Johnson, killer
remember
guy.
you had that on stream too, and I think that was a discussion too about like the ethics behind like
Yeah.
essentially stealing, right? Like unwillingly, right? As some
Uh huh.
celebrities do, versus you know, like the more responsible thing, like how you do, like you have on the background like these 3D printed skulls and fossils that are based on real things. Yeah, exactly.
Yeah, or Allosaurus right here.
And it's a great way to learn, and without, you know, like you always say, it belongs in a museum, and I think that's a really cool way of doing it.
Yeah, thank you. Yeah, I appreciate that perspective from people.
Yeah, absolutely. I mean, we, you know, for us, it's like there's a few of these older pieces we have of the amber, but most of them, yeah, like the really, like, you know, these are like the low priority ones that are here with us that are donated from a lab versus like those really high ones that stay within a lab setting and, you know, are studied or they're shifted to museums, for like the really those rare pieces. And it's important to keep account of those because, I mean, really, like you said, we're kind of, you know, responsible for everyone learning, and what better way to learn than in a museum?
Definitely. Exactly.
Moon Pie was not having it at the end. She's like, nope.
Yeah. Well, you know, that's how cats work sometimes.
Oh yeah, no, you're petting. I keel, and then you pet some more.
Exactly. And then she turns around like nothing happened. Like, oh yeah, I didn't just try and claw you or bite your jugular vein.
Yeah, exactly.
Yeah, it's fine.
Thank you again, Danny, for coming. I love these segments so much, and like chatting science. I love the papers.
Yeah.
Like, I think we've got some good stories. No, sir, thank you. And guys, if anyone's coming in from the front page, we have, I think, 2 days left, or maybe we'll get March. It's not clear. It's not clear what they're gonna pick. But if anyone's coming in the episode description today, there is paleontologizing in 2 places right now. Please click on his name and click the heart button if you have not already. Live fossil science during the summers, like in the field collecting, as well as 5 days a week answering all your fossil questions, viewer-based questions, directed segments as well, occasional video games, art, just a renaissance man is Professor Anduza. Please go drop him that follow.
You make me blush, Balloon. Holy cow.
I speak nothing but the truth, sir. I speak nothing but the truth. What should we get started on? What paper should we start today?
I was kind of thinking maybe we start with one of your papers. There was one of them that I had difficulty parsing because it was a little in the weeds for me. And I feel like I would benefit, and I think the audience would also benefit from a quick overview from you as a geneticist about the DNA transcription and the non-pig one, the non-porcine paper.
The pioneer factors.
Yes. Yeah.
That's so... It... Weird... Like, give you a little background. Hi, Creatrix Brett. Madame, welcome in. So it is called a pioneer factor. And the reason I'm spending a moment on this is because it has been said that the term is controversial of how it's used because there's negative connotations with the word pioneer. It is not meant to be used in any kind of derivative way. And actually, the guy who came up with the term is actually at Penn. I worked down the hall from him, and it's possibly that he's going to be for a Nobel Prize one day. Like, this is how big this kind of discovery is.
So this sort of thing is close to home. Like, this is somebody who lives and works right near you, where you live and work.
Yeah. So he wasn't an author on this paper, but the topic of pioneer proteins is something that like he originated, and you might be like, well, so it must be something cool, right? And what it basically is, I have a figure for us because I like to explain stuff with images. And actually, hold on, I got a better one to start with. That's a little bit, on the easier side of things, and then we'll go on to the more difficult one. There it is. So, in the most basic terms of epigenetics, right, there is this situation that we have where your DNA is folded on itself and is wrapped around in loops. And when it's wrapped around in loops, it's wrapped around this ball that's called a nucleosome.
And this is within the nucleus of each cell. Correct?
Exactly. Because if you were to unwind in one cell of your cells, your DNA, it would be taller than the rock. We can go with the rock description.
Okay, so more than 6 feet, 7 feet, 8 feet, however
as
tall
like
he
a
is.
singular
Okay.
strand, right? And it's like, well,
Uh-huh.
how does that fit in your cells or any cell?
Uh-huh.
And it's by this idea of compaction. So chromatin is
Uh-huh.
wrapped around these what looks like beads on a string, and these beads are called nucleosomes. And so you can see
Okay.
here, this red is the string, and it's wrapped around these yellow beads called the nucleosomes.
Uh-huh.
And so the question is, okay, that means every cell has this, right? Every cell has these nucleosomes. Every cell in your body has every single gene because it has all the DNA.
Sure.
What allows a cell to become a heart cell or a liver cell or a brain cell, right? Because what is the thing that's going to be controlling that? And so this is where this idea of a pioneer factor comes in. During development,
Okay.
there is this very critical enzyme that goes in and essentially hooks itself to the nucleosome. And it will start the process of opening up a particular region of DNA. So it binds. So, like here, it's that blue circle.
Uh-huh.
I'll zoom into the upper one. That is your pioneer factor. And it's called pioneer because it is going into the unknown. There was never something that was known as touching condensed chromatin. And so and the only reason I point out about the use of the word is because I can see it, and it's actually, it's something to keep in mind with science and like talking about the changes in words and what they mean. It might be one of those that will eventually be changed.
They might rechristen it a sorting hat factor or something like that.
Yeah.
Use Harry Potter-esque. I do not know about Harry Potter, but like, does that make sense? Like, it's designated what kind of a cell it will become.
Yeah. And it's not so much, so would be calling it like a master regulator. So like a very high level organizer that helps determine what something becomes. And what's important about that is there's different types. So there's one thing for deriding this type of cell, and there's a different factor for this other cell type. And why this is cool is because it's able to bind to condensed chromatin, which is weird. That is not something that is standard. Normally, like here in this second part, we see these additional transcription factors, these additional TF, and those bind to DNA directly. And that's usually how we turn on a gene. Like that's your standard. Those aren't binding to the yellow blocks. Those are just binding to like this blue region here, or the red right at the DNA. Like that's what they're binding and sitting on. And that's like your normal, what we've known since the 80s kind of deal, even if not earlier. But this, I think the discovery, the original one was like in 2008 maybe, like pretty new, that they identified that there was this binding event happening. And so what it again lets you do is it modifies this nucleosome to shift it. And because when the DNA is wrapped around the nucleosome, you can't turn anything on. It's in this like condensed state, and it's in this off state. And so when you move the nucleosome over, you essentially unfurl it, and you now have access to genes in question.
So it's almost like you've got a fishing reel there, and then like you unspool the line from that, and then you can alter it. But when it's spooled up on that reel, you can't alter it. Is that kind of the idea?
That is a great way of saying it. I've never thought of it as a fishing wheel. That 100% what we have here. Yeah. And so it's you need something to be able to open it up. And so the question I've been for a long time is, well, what is that thing that opens it up? And so the original one was like in a unique cell type where they looked at for differentiation. So like, a neuron becoming a neuron, a heart cell becoming a heart cell. But what it's really cool is it turns out nowadays it's also for other epigenetic factors. So let's say there's a group of genes that are metabolism related, and they're normally off in you, and you are undergoing some kind of starvation stress. So you need to now change whatever metabolism genes that are in your cells, but they might be in this condensed state. And so then you open them up, and so you can then turn on those.
Unspool them, basically, so you can get to a spot where you can change them.
Exactly. So it was classically the first time identified in development. But now it's thought that it's actually like when it's trying to open up these regions of DNA that are closed, it's these factors that are doing it. And they're weird in that they do multiple things, but not everything. So there's not one factor that does everything. It's like there's this one factor that does a couple of different things, and they're rare and they're difficult to identify. But once you get one, you can figure out, okay, what regions are they opening?
Gotcha. And is pioneer factor here a generic term for lots of different factors that can do this, or is that a specific kind of factor?
So it's, I guess, both. It is a specific factor that does the unspooling.
Gotcha. But there can be different kinds of pioneer factors that can...
Yeah. So there'll be one that'll be, this is going to push your cells to differentiate into neurons. And it right, because it's like the mystery puzzle. If you have in your cells all the DNA, all the genes that can make every cell in your body, right? What turns on the right things and keeps the other things off? And that's the hard part. It's like, it's still, even with knowing these factors, it's really, really difficult to tell, like, what's being modified. But it's like the original factor that's actually doing it, which I think is remarkable that we were able to identify something like that.
Very cool.
So that's like the base of it. And so once, the next step is once you've shifted or unspooled that thread. So here, imagine that you take this nucleosome and you've pushed it down. And so now instead of having 3 on the screen, it's not that we've gotten rid of it. It's just we've pushed one a little bit over and like unspooled a little bit. So we've opened up this region. Now you can actually turn on that gene, that blue gene, whatever that may be. You can now turn it on. And what that means is it'll make messenger RNA, which will make protein, which does something in your cells. Whether that be neuronal elements, or it'll be, liver elements, or what have you. It'll turn on those components there.
Interesting. Wow.
And it's weird that this factor, the pioneer factor, does the twofold job, because not only does it act on the nucleosome, that unspooling, but then it also sits a little on the DNA and like recruits the other factors to actually go do the transcription, to turn on the gene, which is really cool. So that's like the base of what one of these is. And Just Corp, welcome in. Professor Anduza is eating a delicious lasagna.
I am very lucky to be doing that. I'm joining Balint for a crossover stream right now, and I've been streaming for like 5 hours before this. So really lucky to be able to eat some dinner while Balint teaches us about genetics and development like this. Balint, this is kind of mind-blowing. As a paleontologist, this is not something that I am familiar with. Like, we learn in school about genes and about different genes switching on and off to cause different, character states and that kind of thing. But I never learned that it was this complicated, or that there's this much nuance and variability. And it's like the more that I learn, the more I realize that I don't know about this kind of thing. And I assume that's probably the case for most people watching this right now. It's complex stuff.
Yeah, well, because this is like 10 level, because that's the same way I had, right? Like when I was in school, it was junk DNA, right? Is what we had. It was like, right? Everything was like,
it's just extra ballast. It's like leftover, doesn't code for anything. Just forget about it. It's junk.
Right? And it's like now we know actually that's really important non-regulating factors and shape of the chromatin and blah, blah, it's like, so the reason I thought this would be a cool study is because it felt like, we've been talking a lot about aging on the crossovers and epigenetics, and this is a potential regulatory mechanism of how things like this might work on a global level. And I just thought it was really cool. And like, there's no reason why this couldn't happen in dinosaurs too. We know in birds, there's also this chromatin structure where if you you have to be able to fit the DNA in a cell, and so you do it like this. Even fruit flies, something quote simple, have chromatin that's also compacted like this and has these factors. So
Yep.
what's Nelf was asking, like the real life applications to medicine, like you can study this in something simple, and it is a very, very complicated thing to learn in something simple. So in humans, like Nelf, I don't want to lie at this point and tell you we're really close. It's so complex that you have to be able to figure out where this pioneer factor will bind and what will it recruit and how will it turn things on, because sometimes, right, if you don't turn it on the right way, it might over recruit and it might turn stuff too far on, and what then happens is anything that's over activated usually is cancer, right? Like that's usually where we get at. And so it's this really fine balancing act of getting something like that right. So it's difficult to get yet, Nelf, but it's moving in that direction. It's primarily studied again right now in the context of development, which I think is the easiest. Again, it's not an easy question, but it's the fundamental of this is a liver cell, this is a neuron. How does the cell know what it to become? And that's that level of regulation. And then Nelf, later on, we have seen that in cancers, there's growth factor regions that are usually in this closed state, but then they get opened up, and then when they're open, the growth genes are turned on, and then out of control cell growth is cancer. And so these factors do a lot of different things, and it's like that again, that master regulator is what we call it, like at the very top of the map, to where like then this thing happens, this thing, this, and you can really nicely trace it back to that original starting point. So that's why I was like, this is really cool, and just as like a more complex image, to like really get at how hard this is.
Wow.
Yeah, let me blow it up a little bit more. When you ask Danny about the different kinds of these pioneer factors, like what are the different other things that they do? So they can act on not just the DNA and the nucleosome, but what the nucleosome has modifications on. So sometimes you can open up the nucleosome with a certain kind of modification, and sometimes you can close it with different kinds of modifications. So there's what's called the H3K4 methylation, and what that means is there is an amino acid that is identified with the letter K, and its third hydrogen, or it has 3 hydrogens put on top of it, and that puts it in this closed state. So this is like a chemical readout of a modification that's done on it.
Okay.
And so there's different kinds of modifications that could be done that help like to differentially close the DNA. So NMD, a nucleosome, is a ball that if your DNA's string is wrapped around to allow it to fit in your body, in one of your cells. And yes, the question from earlier in the chat from Timmers, if you pull out a DNA strand from your cell, it's larger than 6 feet. From one cell, if you pull out that string, it's over 6 feet. One cell.
It'd be incredibly thin though, like just so much thinner than like a strand of the thinnest fishing line or something.
Yeah, like very, very thin, but still it's how does it fit? And the way it fits is you wrap the DNA around these beads, and that really, really compacts it. But then the question in NMD is, well, how do you get to a region of DNA to turn it on? And so then you have these pioneer factors that shift the little beads around to open stuff up. So Timmers, you were asking about, well, how does a cell know what it needs to be? There's some modifications done on these nucleosomes that are specific to, let's say, your liver cell. And there's other kinds that are more general for neurons. And so if you have pioneer factors that act on this kind of methylation versus a different kind of methylation, that's that way it makes it very tissue specific. So they are able to turn on particular elements in a given tissue, and they get to turn on the right things. So this paper is trying to identify one of these factors for a very particular question. Like, what exactly is being targeted and turning on during this kind of like reprogramming state of something like that. So it's in here it's for fertilized eggs. So this is like the paper for today is like, imagine very early in development where you just need a fertilized embryo to divide and make cells and like everything being on, because when that embryo, right, is unfertilized, everything is in a very compact state. Like there is not in a human embryo or a mouse embryo, just like the egg, you're not having loose DNA everywhere.
Right.
Because that actually, Timmer's loose DNA is easier for mutation to hit.
Huh.
And actually, Danny, this is like, so I'm sure you've heard where it's like older moms, like once you hit 40, there's this higher likelihood of there being mutation and trisomy in babies.
Is it because the DNA is looser like
in part, that element does happen, yeah, because it's apparently like, and I guess this makes sense. If everything's super tight, you can't really break something, or if it does, it kind of just links back together, right? Because it's like by its neighbor. But if it's a little bit looser, you can imagine a situation where actually that's how we get Fragile X, where part of the X chromosome, the very tip of it, breaks off.
Oh, really?
Actually, I can find a picture
of that. That's like more simple than I would imagine. Like it, which is really interesting because so much of this stuff seems so much more complex than I would imagine, and yet certain things are like, oh, really? That's the answer. That's like simpler than I would have thought.
It's like simple from the perspective, I think, of like conceptually, but then it's like,
yeah.
But then how do you fix it, right? Because that's where the complexity, I think, really comes in. It's like you can see here on the tip of that chromosome, there's just like this little nub.
Yeah.
Oh, come on. That blow up was terrible.
Control minus to make it bigger.
I know, it's...
Could you do open in a new tab, maybe?
Yeah.
Control, or a right click and then...
It loses the res, and oh no, it's gonna do it again. Oh no, here we go. Alright.
Nice.
So yeah, like, there's that break point, and you can see it's like clearly separated away, and it's because instead of being in that tight, compact state like the rest of the chromosome, there was some loosening over time, and now you've acquired this, like, it did break, and it's still like repaired and attached, but it's not quite right. You know, it like looks
kind of herniated out almost.
Yeah. And that region of it, so this actually comes with a lot of genetic disorders, right? Because the X chromosome, it's really difficult to compensate for because there's this issue of dosage compensation where, you know, in genetically male, you have one X and one Y, right? And so the X, just everything's on in that one X chromosome. But in a female, you have two X's, and if you have all the genes on, if every gene was on both, you would get double dosed and you would die as an embryo. So it's wild. In genetically female with the XX, there's selective turning off of genes. And it's not all on one chromosome. It's like, of these two genes, like it might be this chromosome that's turned off, and then this other one's turned off on the other chromosome, and that's how you regulate this important dosage during development. Because you can imagine if you overdose on a gene, the embryo, it's lethal.
Okay.
And that's even in like fruit flies too. It's the same kind of component, like this dosage compensation. And so here, those genes, like we're messed up on those genes, right? Like there's like the accessibilities there. Even if stuff was turned on, like there's inversions and like the genes have turned around positions, and you're not actually turning on things that are meant to be turned on because they're broken.
Yeah.
And so in part
when you're talking about genetically female versus genetically male embryos, in a genetically female embryo, if one of the X chromosomes has Fragile X, that's fatal to the embryo, but it's not necessarily for an XY embryo? I don't understand that. So
patients will be born, and so it's actually, I believe, let me double check real quick. I'm gonna hide the view so that nothing triggering comes up. I believe it's male. It's
Sure.
fatal.
Oh, in males it's fatal, but not in females. See, that would make intuitive sense to me, because it's like if you've got 2 X's, then you've got some wiggle room, but like 1 X where something goes wrong, that seems much more severe to me.
Yeah, okay, so let's see. So there are males and females born. It seems like with both. But
Okay.
it looks like males have a shorter lifespan.
Oh, that makes a lot of sense.
Okay. And it looks it's actually, so there's a it's weird of how it presents, because there's physical and mental, and it's weird to me because it's only one chromosome that's perturbed in like one region of it, and so it's kind of weird to think about, well, everything else is normal, this is just this one region of the chromosome, why does that add so much? And it's just kind of shows you why the dosages on some of these genes are just really, they're so tightly regulated that any misregulation yields something really negative. Yeah, if anyone's interested, I won't pull it up because, you know, it can be a very depressing topic, but there is just a quick wiki on it, and this is a study here of tracking males over time, if anyone's interested. So this is like looking at 34 males with Fragile X and seeing like clinically how they're like what happens over time with them in like a lifespan study.
Interesting. Wow, this is harrowing stuff.
So yeah, for these factors, why this is so cool for this new paper. So this paper came out 2 days ago. So like this is like a very new cutting edge paper, and the whole point is they were trying to find out, like, in that egg, in the embryo. This isn't like anything sexed yet. This is just in an egg that's like not yet fertilized, right? Everything is really compact. When it does get fertilized, it starts dividing and growing really rapidly. And what is the factor that turns these genes on? And so they said there must have been a pioneer factor, because you go from everything closed to a lot of stuff open. And like, how does that work? And so I like this because there was this, they did this technique called, actually I've got the process as well in an image, cryo scanning electron microscopy. And so essentially it's a very powerful way of looking under the microscope. It's a very unique piece of microscopy tool. It actually involves force of dropping a cell or a particular molecule, and as it's dropping through this electron microscope, I don't know if you can see the arrows going down, it's dropping vertically, and then it's in as it starts to drop, it gets frozen, and then it gets sliced, and then you get this multiple images from multiple angles. And so the idea is you make a 3D structure of it.
Sure. Okay.
And so what the researchers wanted to find out was, what is the molecule that is doing this transcription in an embryo? And what does it look like? Where does it bind? And apparently, so there are tricks sometimes to be able to ask where something is binding. There is apparently a very cool genetic trick to do, and I've actually done the classic genetic trick for it, which I just wanted to show y'all real quick how you can normally do something like this and why you can't do it here. This kind of will, I think, illustrate why this is a neat technique, where if, let's say you have a region of DNA just on that line, and you know, there's something that binds right here, what you want to figure out is, okay, what is that thing and where is it binding? Right? And so what you're able to do is there's a protocol called fixing. So you put the DNA strand in formaldehyde, and what that does is it links everything bound to the DNA, it's just stuck. It's permanently stuck. And then what you do is you give the DNA, onto the DNA you put a molecule called an MNase.
Mm-hmm.
And what that does is it basically chops up any open region of DNA. But the part where your molecule, your secret molecule is bound, is protected. Because it can't be chewed up because it is covering that region.
Okay.
And so you break down everything else, and you're left with your molecule in question holding on to some DNA, and then in your vial you separate the two out, and then you can identify what the sequence is that it's being bound to and what the molecule is. Like, it's super elegant.
Huh.
Not always easy, but like relatively speaking, it's a good.
It's got a nice simplicity to it though, like elegant, like you said.
Yeah. But it turns out this doesn't work on something that's really, really tightly bound, like the chromatin.
Huh.
Because chromatin that's tightly bound inherently does not get chopped up by this MNase, because it is almost self-protecting. Like, it is so tightly bound that there's not just free strands of DNA floating around. And so you need this extra way of actually figuring out what is being bound and how to actually address that.
Right.
Yeah. And travel, thank you for the stress travel world.
This is interesting stuff, Bullen, because like I'm not, as a paleontologist, somebody who knows next to nothing about DNA. I used to think, DNA and genes and all this genetic stuff as being very kind of abstract and abstruse, and not like a tactile thing where like, oh, certain parts get stretched out and certain parts are too tightly bound for other things to bind to. It makes it feel, the way that you describe it really helps, because it makes it feel like, I don't know, a Lego set or something like that you can actually like put your hands on, and it feels real and tactile in a way that I'm not used to thinking of these things. I'm used to thinking of them as abstraction.
It almost feels like a slinky.
Yeah.
Where if you have, I don't know if you ever had like the multicolored slinky, like the rainbow one, where it was like, red at one end and violet on the other, and when you have it close together, you can't pick out the region of color. But if you open it up, you can then pick out and like run your finger on the green. And I guess that's kind of it. Like, the transcription factor that's turning on a gene is where you're rubbing your finger that's green.
Sure. Okay.
But when you close everything together, you can't actually tell me like which one is green and where's the boundary of green and blue and all these, because they're just so tightly backed.
Yeah. That makes
so
sense.
Yeah, that's why this method needed to be done. And it's a really cool method because you get the 3D structure of it, and this is used for other stuff too. This cryo EM, so it's still electron microscopy, but it's cryo because it's you're freezing something.
It's cold.
And that, exactly.
Yeah.
And that you're
like Cryolophosaurus, the frozen crested lizard.
Yeah, there you go. Where was one more time?
Cryolophosaurus.
Where was Cryolophosaurus?
Take a guess, Blinn. What's the coldest of the continents on our planet Earth?
All right, it's not Antarctica, is it? There are dinosaurs, but at the time, was it cold?
It was warmer.
It was the Pangeal geography.
Yeah, so Antarctica would have been a little bit further north. It still would have been cold, but there were ferns and palms, and well, not palms yet, I guess, in the early Jurassic, but ferns and horsetails and other like subtropical plants that grew back then. Yeah. Anyway, I don't mean to sidetrack us, but yeah, there are dinosaurs from all 7 continents, including Antarctica.
So,
yes,
we're totally rabbit holing. How hard is it to excavate something in Antarctica? I imagine it would be.
It's hard to even get there. Yeah, so like I have, one of my dreams is to do field work in Antarctica, because chances are if you find dinosaur fossils in Antarctica, they will be new. They will be a new genus or species, and that's really exciting. And also Antarctica, like, oh, this whole continent for science. It's a very like romantic notion, but it's extraordinarily difficult to dig there. Like to find areas of exposed rock that are not covered by ice or glaciers. From what I understand, basically what their typical method is to like prospect along and look for bones that are exposed in whatever rocks are exposed above the ice, and then they literally saw them out with rock saws, and then just bring these giant slabs of rock back to base, and then back to laboratories on other continents, and then they try and extract the bones from the rock there. So it's a very different way of like our typical fieldwork methods of like using plaster of Paris and burlap to like jacket fossils. They don't do that there. Everything would freeze. So they've got to basically just saw out giant chunks of rock and then carry them back.
Wow. So it's, I guess what also makes me like wow about that is that because you told us like when you're walking around and doing field work, like it makes sense to me of like, okay, if you see a dino bone sticking out, right? You have a search image, you know if you need to dig there, but everything's exposed like that. Like you're not covered in snow and ice, and so it seems like you're even more limited if you're searching in the Antarctic, right? Because like the likelihood of something being exposed versus like, you know, under 10 feet of ice.
They normally dig during Antarctic summer, which would be winter in our northern hemisphere. So like they'll be down there in, you know, November, December, January, where it's summer in the southern hemisphere, and that's when you've got the least amount of ice and snow covering things. But still, you got to find particular exposures of rock that are not covered by ice in order to be able to actually prospect and find those fossils.
Yeah, so that seems really difficult to me. If you do come across an Antarctica paper, could we do that one week?
Oh yeah, you bet. Yeah, we've looked at some videos of Antarctic field work in the past on my channel. But yeah, they're kind of few and far between. There are definitely the fewest dinosaur fossils known from Antarctica out of all 7 continents. They're not common from there. But there's like 3 or 4, maybe 5 different dinosaur genera that have been named from Antarctica at this point. Cryolophosaurus, Glacialosaurus, Antarctopelta, and then there's a Dromaeosaur whose name I forget, and I think there's another critter. So I think there's 5, there could be a few more. But not very many.
Did you say Glacialosaurus?
Yeah.
I love that.
Yeah, that was the first dinosaur, I think, to be found in Antarctica, but not the first one named. Cryolophosaurus was found later, but named first. It looks kind of like Allosaurus, kind of like this guy here. Except it's got this like cool, almost like comb-shaped crest on the top of its head, like that. And it was a big deal back in the 90s. It was in a lot of like new children's dinosaur books and stuff, and they nicknamed it Elvis at the time because it had like a unique like hairstyle thing going on with its crest. But anyway, it's a really cool critter.
I love the naming system for it. Like it was going with like ice themed names. Imagine it'd be a gold mine for discovery, right? Because of like the isolation presumably that happened, and then you want to wonder what cold adaptations might have happened, or like when they died out in relation to like how the ecosystem was changing. And I know there's like, you know, insects there too that are still alive today that have almost antifreeze like properties in their blood. And that's something that is studied.
Like the Weta, I think, from New Zealand, right, where they can like freeze solid, and still survive. Yeah.
But there's these Arctic midges that instead of freezing, they're just active. And like that's
one way to do it.
Yeah, like that's all. Like it's because most other things, right? You were saying would freeze their bodies, and then when it's time to warm up, they'd unfreeze. But these are able to year-round survive at that temperature, and it's like, wow, that's really cool. And so I think it's like really you said, like the romance of that country or like the continent is really exciting to think about, like what potential things could be there.
It really is. I would love to do field work in Antarctica someday, but you got to know the right people, and I've heard it's almost like training for a space mission, where it's like you've got to jump through all these hoops to be able to prove that you can survive in a situation like that, because it's not just physical hardship, but also like mental hardship. You've got to prove that you can be like a cohesive member of a team and not get into fights with people and stuff because you're isolated, and it's a lot. So like there's a certain like almost esprit de corps, or like an elite feeling where it's like if you could do field work in Antarctica, that means you're like in the creme de la creme of field researchers, you know.
It's like Danny, have you seen this video on the biodome in Arizona?
Yeah. Or I haven't seen the video, but I'm aware of it. Yeah.
Yeah,
Yeah.
there was this. Our friend Cornrellis showed us this video about like there was an attempt to train people in that ecosystem, right, to lock them away and
Yep.
see if they can survive for a year. And that
Uh-huh.
almost comes to the similarity of what you're saying. Like if you have the wherewithal to go to Antarctica,
Very similar.
that ability to
Yeah.
work together, and it failed the first time because everyone bickered and fought.
Yep.
And then
Yep,
there's
'cause
also
those
like
are tough
problems with the
conditions,
building,
just
but
being crammed in cheek by jowl with other people, and like there's just conflict that arises when you're that cramped in. Yeah. Yeah.
and so that's
Yeah.
just what it reminds me of too. Like you were saying, it's not just as simple as being sent down and you can just be there and everything's fine. It's like there's these other human considerations to think about that maybe aren't top of mind when it comes to something like this. And yeah, Reiko,
Yep.
I haven't seen the documentary. I learned about this from Corn Relish like a few months ago with this video.
Rico Dactylis is making a joke. That was a comedy movie, notoriously bad with Pauly Shore.
Oh, see,
Yeah.
I mean, I was, I'm like, I know Pauly Shore. Like, all right, I guess he was in that kind of movie.
Yeah.
All right, well, sure. Well, several people were saying it, and I'm like, all right, sure, Pauly Shore was in a document. All right, cool, I'm okay with that.
I've never seen it, but I've heard bad things. Yeah.
Y'all got me. Y'all okay? Y'all got no. Claire. We don't say sorry here for the silliness. Oh, you used the kappa. Okay, fair. I had to almost delete Claire there for a second because like getting no apologies on this. But so yeah, I guess in this tech they're using this ice and they're taking these like really thin slices, right, to get this 3D map that they're putting together. And this technique can be used for a lot of different things. It's very, very expensive to do, as one might imagine. Like the electron microscopy in general is stupid expensive. Like the one machine is over $1 million. Yeah, just the microscope, not the service contract or anything is $1 million.
Oh man. Oh.
And
Yeah.
to me, where it's even wilder on that front is like not only is it $1 million, but if you don't buy the service contract and something breaks,
They come and break your knees.
oh yeah, like there's a machine. we talked about like the mass spectrometer that looks at odorant molecules and other small molecules like within a cell, even like what's bound to DNA, what's not, etc.
Yeah.
Fortified. Yeah, Fortified. First time chatter. Welcome in. Thank you for that follow. That machine, a mass spectrometer costs $400,000. Every year is a $50,000 service contract. If something breaks, which
Gotta pay protection money.
70,000. So the lab that was next door to us bought a mass spec, and they're like, we don't want to spend $50K a year on a service contract, and like a critical part broke because what it does, it aerosolizes the molecules and then magnetizes them to blow into the machine so that everything's individually like pulled out, and that part broke, and that was $70K. And they're like, well, we should have gotten a service contract, and he's like it's almost cheaper to...
if you didn't have a service contract.
Exactly. It's like, wasn't that like a Monty Python thing? It was like there were mobsters show up to the military and be like, "Oh, it's nice tanks you got there." It'd be like a shame if something happened to them.
That sounds like Monty Python. Yeah.
And it's just
yeah.
it's crazy how expensive the equipment is, but that's what that's not that it makes sense. It's just that you're doing something really cutting edge here of getting the 3D shape of a molecule.
There's probably, how many of those devices are there in the world? There's probably fewer than a dozen, right?
Cryo-EM is becoming more popular. Like usually for the electron microscopes, maybe a university will have one if it's a wealthy university. Thinking like an R1 level, meaning that's like the high tier for what funding they can bring in. there might be one of each. Because actually a transmission electron microscope for slicing through a cell and a scanning one are two different ones. So then you're talking, a cool $2 million to have the set. That's like a big university. Like your smaller schools, there's no way they can bring in the grant to get something like that. And then a cryo-EM is an additional new piece of equipment that you have to buy and set up. And so I don't think it's just like 12 in the world, but you're definitely like 50, I would say. Like not a lot. And it's like
so it's like the same number of like maybe international airports you might have in a given country. There might also be the number of cryo-EM devices.
It's becoming cheaper as time goes on, but it's still like it's still not cheap, but it's at least like the pricing and pieces are eventually coming down, and people are making innovations to it that make it easier to, I'm not trying to be jaded and be like it's actually cheap. Like we've talked about how much field work funding you get for a paleontological dig. Like this is, that could fund years.
It's like a couple $100 maybe, That would maybe buy you the little rubber feet for the underside of this, underside of the machine. For the amount of money that we get to dig up a dinosaur every year.
Yeah.
Oh boy. Yeah.
It's actually really disheartening when we've talked about like how little paleontology funding getting, and if it just
our budget was $500 this past year, Berlin, for working in Utah. $500 was our budget. Yeah.
What do you even do with that? $500. Jaded, what?
We buy food.
I know, but it's like equipment. Like,
yeah, we don't have money for equipment. We've got to reuse the equipment that we've had for 20 years. Yeah.
It's super frustrating.
Bring our toothbrushes from home for brushing the dust away.
And then you brush your teeth too.
Hopefully everybody's got 2 toothbrushes, but
yeah, it's no, I agree. It is very frustrating to see, but these are like so super special. I'm not making an excuse for the machines, but they're rare, and what the article did was they actually got the molecule. So it's like this figure is really cool because they identify like this is your DNA strand here in blue, in red, yellow, green, and purple. So the inner bit is that ball, that bead that the DNA is wrapped around.
Okay.
So instead of, I'll zoom in here. We'll make it
I'm following so far. This is good.
So instead of like here, instead of just this yellow ball, let's make it more complex, and I'm going to tell you there's multiple regions of this ball. It's still a ball. It's just there's different regions for it. And so those different regions are different colors here because they have slightly different structures. And then what they found was this region in red, that's that pioneer factor. That's that molecule binding to the DNA.
Okay. Huh.
And so it's...
No, I do have a question here, not to derail. This illustration that we have right here, showing it more complex than the other one that just looked like a yellow yo-yo with red string around it. Is this based on like microphotography or nanophotography, or is this more of an illustration? It looks more illustration-y.
This is the true structure of what you're pulling out. So when the DNA is wrapped around this nucleosome, this is with that cryo-EM. So you're getting true structural information.
Gotcha.
Early on, it was like in the textbooks now, instead of that blue circle.
Together, we can rule the galaxy.
Bojangle Jenkins.
Bojangle_Jenkins just subscribed.
The TTS does not
nice.
enjoy some of the names. Bojangle. Do you know about the restaurant Bojangles in the South? I hope you're named after that. Bojangles, thank you for...
I don't know about that.
It's... Lita thought I was making it up because it sounds like, surely, that can't be a restaurant.
Yeah, like when I first heard about Piggly Wiggly, I'm like, dude, you're making this. That's not real. Well, you're pulling my leg.
Well, and the mascot's a
Piggly Wiggly
pig
is
that's a
built in.
butcher.
Huh?
The mascot of Piggly Wiggly is a pig that's a butcher.
That's a little more morbid.
Yeah, like Shirley, and as Cormeller says, "Don't call me Shirley."
Who's
That's
that
a joke.
pig butchering? I don't know which possibility is the more unsettling one.
Right,
Is
and
it his fellow pigs, or is he turning the tables? You know?
yeah.
Oh boy.
I don't. Right, but it sounds like a joke, and sure enough, it's not. Bojangle, thank you for the sub. A piece of raw science with your name on it. Into the beaker we go, and another one of those partner plus points. Danny, we did complete the first month. We got the first month completed.
Congratulations to the 300, right?
Yes,
300
sir.
points?
Last night
Very
we completed
nice,
it.
very
I looked at the dashboard. I was like, 249, and last night the party people were like, all right, let's make this happen, and just like pushed it over the line. So yeah, we're now at 325. Thank you very much, Bojangle.
Fantastic. I'm very glad to be part of this as well. I think I contributed 6 of those points with that tier 3 sub. Oh man, that feels good to have that goal completed. That's so exciting. Just 2 more months, right?
Yeah, just
And you
2
got the
more
partner
months.
plus? Congrats, Belen.
Thank you, sir.
Congratulations.
And Bojangle
And
says,
congratulations to chat. Very nice.
oh yeah, the chat was like, we got this. I'm like, all right. I was, Lita and I were just like, we're not big streamers. We're not, it's a dream, but we'll get the 60-40 with the 100. And they were like, nope. Chat's like, we're making this happen.
Oh yeah. Oh yeah. Very nice. That's awesome. Holy cow.
And Fishman, sleep well. Thank you again for the raid earlier tonight. Bojangle says, I am from Mississippi, definitely named after the sweet, sweet chicken and my cousin Leroy. All right. Well, Bojangle, I love Leroy Jenkins, even though he did, destroy an entire, video game, mission, and then I do love chicken, too, so you're excellent.
Even I know that reference.
Oh,
boy.
Did you know that was staged?
It makes sense, actually, come to think of it. Yeah.
I was so disappointed.
Perfect.
I was so disappointed that it was staged. I'm like, I really wanted that guy to just have gone off the rails and just run into a place he shouldn't have been.
Uh-huh. No. Yeah, never meet your heroes and never look into the origin of video game memes, I guess.
That's not true, because I've met my hero and you were wonderful at TwitchCon.
Likewise.
See, chat, I won't let him get away with it. I got him.
No, by the way, Belit, we gotta look into, I don't know when the call for content went out last year, but we gotta look into that for, because it's gonna be like a month early this year, right?
Yeah, and I feel like
November.
this is when we met last year to discuss our idea. And
Yeah. So let's talk about that sometime soon.
Yeah.
And yeah. Do another science panel this year at TwitchCon.
Maybe they can put us not on the last day in the last time slot.
That would be nice.
We can be like, hey, we sold out the room, okay? Put us in a bigger room.
There you go. Yes. Yeah. Standing room only, practically.
Oh,
Yeah.
I mean, yeah, I think we actually did have people standing in the back.
We did. We
Yeah.
did indeed have people standing in the back. So,
Super humbling,
yeah.
too. Like, that there's that many people that there's not open chairs.
So,
Like-
and we've got video evidence of that too, I believe. where
Oh,
this
well,
photograph
and
gets
Lordy
itself.
Lordy took some really cool photos from the back that had showed, like, the how... I'm like, this is insane. Because, when you're up there, I don't know, like, kind of... I don't know. I don't look at anyone, like, it's just, like, looking around, but, like, not really focusing on anything because you're, like, in the moment. But, like, wow, there's a lot of people there.
Yep. This year, let's make it twice as big. Yeah.
I think we can do it. I have faith.
Yeah, I think so too.
But yeah, so to answer your question, Danny, the classic image is that yellow ball that we just looked at. Part 2 of the classic image is the yellow ball cut into 4 quadrants. So it has 4 different colors, and that was from like biochemical assays where you can mutate it. It's 4 genes that are governing essentially those 4 different regions, like this kind of nucleosome part, and this is a part, and this is a part, and those come together to make the ball. So if you get rid of one of them, it falls apart. The ball does, and so you can figure out that it's in different parts. And then there are multiple of these cryo-EM images, and even I think transmission electron images where you do this very thin slice. You can see the beads on a string, and you can almost make out regions as well. This is like a much higher resolution image though that you're able to get. So this isn't just like a colored like artistic representation, but rather like this is what it actually does look like, which is really cool to be able to get.
Okay.
And they got it from like there's
Cool.
multiple camera angles and like an absurd number of images that are taken for this too. So it's like
So this is just kind of like an optimized view right here.
it's the average.
To be as instructive as possible. Yeah.
Yeah.
Okay. That makes sense.
So it's
I like that.
I guess like on this figure here, like when they're doing the 3D classification, right? It's all these shapes. Thank you, JD, for the ooh. They pull together all these averages, and so see like on one of them, there's a lot missing, and on another one there might be a lot missing, but it's all those pieces together that give you that 3D like topological, almost looking like a geographical like mountain range map that we're looking at, right? Like the overhead.
Yeah. Or kind of, you know what it reminds me of, and hopefully this doesn't mean that I'm really hungry right now. I don't feel that hungry. Looks like a cruller donut. You know what
It
I'm talking about?
does, and that is Lita's favorite donut. Whenever we go to Dunkin' Donuts, that is, if they don't, like, all right, we might as well turn around if they don't have it in there. That is her absolute favorite donut.
All right. Wow. Yeah. Well, what's your favorite donut, Belinda?
I'm a Boston cream kind of guy.
East Coast. Yeah.
What
okay,
about you?
Okay. I am. Well, this morning I actually had 2 donuts for breakfast. I splurged this morning. And I ate them at the beach. Typical California morning, you know. That's how
That sounds
how it always
amazing.
goes here. It was pretty great. Yeah, Lordy was there too. She didn't have any donuts. She had a bagel for breakfast. But I had a maple bar and a lemon filled donut with like powdered sugar on top.
Yep.
Excellent. Yeah.
We have those here too. I think as a kid it was like the rare thing was a Boston cream. I was like, oh man, that was like the big thing.
Okay.
My dad really liked the powdered sugar with like the lemon filling in there too. But sometimes there was a different kind. It was also it's also yellow, but I think it's like a vanilla base, right? And so he would buy the lemon one, but they would mix it up, and so you bite into it, and then it wasn't the lemon, and you're like, what is this? Like you're not expecting it.
It's a bait and switch.
Yeah.
Yeah. Shoot. Yeah. Yeah, and Lordy is in the chat there. Lordy, they didn't have any lemon-filled ones the other day. I think the other day I got a maple bar and a pink donut with sprinkles because I wanted to share that with you, Lordy. That's why I didn't. Yeah. Anyway, wanted to go for a more generic donut that everybody would like. You know, nobody will refuse a pink donut with sprinkles. Might not be anybody's favorite, but everybody will eat it.
That is very true. You'll never have that left over if you give people the option. It's always the safe choice to go. It's not going to be too sweet. It'll be just fine. Like, it'll be...
Yep. Well, what do you think the leftover donuts are? Because I think maybe like a plain glazed donut. I might be left over. Maybe like a bear claw or something like kind of specialized, highly derived like that, I suppose.
The blueberry.
I don't know what... Blueberry? I don't even... Is that a thing?
So, like, they have the munchkins as well in Dunkin' Donut, where it's like the donut holes, and it's like a glaze with blueberry. I feel like those are always, like, the last one. Like, anything fruit-based is, like, on the tail end. Or, you know, the raspberry filling with a sugar dusting seems like it's pretty... At least the places I've gone seem to be like it's a late one to go.
I can see that too, yeah. I like those myself, but it's not necessarily the first one that I would pick. So...
Exactly. It's like, it's not bad, but if you can only get one, it's not going to be
Yeah, it's nobody's first choice, I guess. Or it's very few people's first choice. You probably will have a few raspberry specialists, but they are not they're few and far between.
I think it's because it's an unknown, right? It could be either really sweet, it could be
Yeah.
tart and
Very
anything in
tart.
between. But
Yeah.
the one with the pink glaze and the sprinkles, it's almost the same every time.
There you go. You know what to expect. It's a safe bet.
Yeah.
Yeah. Yeah. I'm glad we talked about this. I
Yeah.
feel like we
That's already.
made this make sense. Yeah.
I just love the fact that you were at the beach this morning having a donut. That's beautiful.
Oh yeah. Yeah. I used to do that all the time. After this move, not as often, so it's a rarer treat now, but it's really nice. Yeah.
Yeah, that sounds like the perfect morning. Because I bet it's not too hot either. It's just like a nice.
No, it was beautiful. Yeah. Yeah. It was good stuff. It was good stuff.
Nice.
Let's see some cool birds. It's always a good time at the beach. Yeah.
Yeah, so I guess just like on the paper itself, like that's like the big crux of it is trying to identify what this molecule is. This is the stylized one that you were talking about, Danny, too. Like this is like your classic beta coils, alpha sheets, right? Like what the structures are like, the classical structures that are in here, and like these are very much like the recreations that we know, and the colors also mean different things. Like the darker colors are like the binding sites that are more likely to get bound to, versus the lighter blue ones are less likely to be bound to. So there's also probability superimposed on these models, and I think it's really cool to see all these little extra elements to it. But yeah, so they found that there was a molecule called this NR5A2, and it's almost a new class of pioneer factor. They called it the totipotent pioneer factor because it's starting from the embryonic stage where it can become anything. And so that's just like a higher order name for a stem cell that still has the potential to be anything in the world.
Gotcha.
And then as you move down and specialize further and further, you lose that totipotency, and then you could become pluripotent, and then you become derived specialized cells. And you can revert like a skin cell to a pluripotent stem cell, but you can't ever revert it to a totipotent base start where it can very easily become anything.
Gotcha. So to go back to our donuts analogy, it's like your basic dough donut before you have any kind of chocolate or maple glaze or sprinkles or anything. Before it becomes a cake donut or any of that, it's like just your basic round shape. That's like your totipotent one, you called it?
Yeah, or I guess if we have let's take that Boston cream. That's your specialized cell.
Sure.
You can revert it to a stem cell by scraping off the frosting off the top. So it's mostly reverted, but you can never take out the filling and make it into a totipotent like the very beginning stage.
Gotcha.
At least not yet.
Uh-huh.
It's just that it's like there's enough derivation and specialization in the next step that it's very difficult to further push back something.
That makes sense. That makes a lot of sense.
See, this is why, chat, we talked about donuts. This is all planned out, clearly.
Yep.
But yeah, so that's this like beginning stage is what pushes the cell to become a certain component or like to start growing. And what they found is it goes into a particular region of the DNA. So there's the major groove and the minor groove of the DNA, which is something that we like. It's always in school, but like, is it something we ever focus on, right? So like,
yeah.
It becomes a really easy thing once you see the picture. Here we go. Open image. All right, so because it's when you're coiling DNA, it's not it's not a repeating coil. There's regions that are a little bit wider, and then it's followed by a shorter region, and it's a wider region and shorter region. And so that's just because of how
perfectly consistent. You've got certain areas that are squished together more than others.
Well, so it's alternating. So you have a wider bit and then a thinner and wider and thinner, and that's just because of the structure, like what chemicals are like your base pairs and how they're built. That's just like your standard
Okay,
instead of having like a standard coil everywhere. It's this like very unique structure built in, and so there's this major groove, which is the bigger one, and the minor groove, which is the smaller one. And so you can, I think, kind of see it on this image. There's one that's a little bit more spaced out, and a one that's a little bit closer. And they found that this Pioneer factor binds to the cramped version, like the cramped part of the DNA, versus
so if you were to take this and look at the backside, would the green part, the minor groove, be the major groove on the other side, or is the major groove still the major groove on the other side?
It is still the major groove, but it is hard. You can't easily tell. So like you have to look at this like front view, because that's where you're seeing like where the opening is located. So like it's
gotcha.
You look on the back side of this, you'd start to see these hydrocarbons like pulled in.
Yeah.
And it would still have a difference in width, but it's not as visibly apparent. I wonder if there's an image where we can. Maybe this one's a better one to actually further visualize. No, I don't want lecture notes. Hold on. Yeah, this one should be fine. So
on the right there, yeah.
Yeah, so here, this is your minor groove right here. This smaller one. So you can kind of see how the other, like it's see-through of how it would look, and then you've got the major groove here as well, and then it corresponds to like where it would look at on this component. So like here you've got a major groove, and then you've got the minor groove here, which is the smaller one, which would be located here. So it's like, I think that's maybe a good image to look at and see. So yeah,
that makes a lot of sense.
It's just trying to identify where exactly these factors are binding in the very early stage of development. And Kramish, thank you for the follow. Welcome in. And so I thought it was really cool because it gets at something that we don't regularly hear about, which is these Pioneer factors, and then primarily because they're so hard to study. Like you saw, you needed really specialized equipment. Like that cryo-EM is no joke. It's also, there is this other sequencing that they did called, depends on what school of thought you're wanting to be part of. It's either ATAC or ATTAC sequencing.
I like ATTAC sequencing.
So the creator called it ATTAC, but everyone under the sun that I've ever met says it's ATAC. This is like the GIF and the GIF argument all over again, right?
I say GIF. What do you say?
I say GIF because I never heard it. Like I read it the first time and I saw it I was like, oh, that's what the English phonetics, surely. And it is totally, it is GIF. Like that is the correct way of saying it, right?
I think it is, but I'm not going to make that argument and say that I'm right. I don't know. Tomato, potato, to each their own.
Exactly, yeah.
But, yeah.
And
anyway, really interesting paper. How, as someone who's actually in this field, Belen, give us some perspective on, like, how significant do you feel this paper is, and, like, how profound are its implications, and, like, what kind of is this high profile? Is this a high profile journal? Are these authors that are very well known? Put this in context for us, I guess.
Yeah, so it's in the journal Nature Structural and Molecular Biology. So it is in the Nature publishing family, which is big, but the big paper there would be Nature. And so
right. So this is almost like a suburb of the journal Nature or something.
Yeah.
It's smaller time. It's more specialized.
It's not like going down to a journal called like Parasitology, which I've published in Parasitology. I enjoy that journal, but that is like highly, highly specialized and doesn't get cited a lot, so the impact factor is low and the whole publishing issue. Versus these are still above what a mid-tier publication would be, because they do have to have, like, there's still a strong rigor to get in.
Yeah.
But it is specialized. So, like, when you said what's the implication of this, I think this is why it went to Nature Structural Biology versus Nature, is because we don't have an implication for it, right? It was...
Gotcha. So I'm trying to, like, analogize this. Would this be the equivalent of like Proc B, like Proceedings of the Royal Society B or something like that, or would this be higher or lower?
A little bit higher. A little bit higher.
A little bit higher?
Yeah.
Higher than Proc B? Wow. Okay.
Yeah.
I'm glad I asked. Yeah.
Yeah, so it was that, but that's what's getting me is like they did try to do a little bit of functional work of showing like what exactly is the result, but it's very thin on that. It's much more of like, here is the structure, we identified it, this is what it looks like, this is the place that it binds, but I don't see any repercussions, right? Of like what happens if you mutate it, if you change its expression level. Like, yes, it binds the chromatin and it opens it up, but like, you know, I mean, obviously you wouldn't do it in a human embryo, but you could do mouse embryos, and because it's the same factors, and figure out like what is the. You don't let it come to term, but you just see like what genes are turned on and off, how that affects the development in the first few days of the organism, like those kinds of questions. I think that's the difference, right? So we had in chat, you know, a question of like, could this cure blindness or something along those lines, like of a cure for something. I think it's too early to do any kind of cure anything.
Sure.
Because it's like, it's our first big step. Like, identifying it is hard. Identifying where it binds is also hard because of the special nature of this protein. But now it's the functional part. What exactly is it doing, and why is that interesting? Like I think it's interesting from a intellectual perspective, but the next step being, you know, like, what exactly is it doing? If I turn it on too hard or turn it down too much, like, what are the results going to be? And there, you know, I think for blindness, I think this wouldn't work because I think it's very general, right? It's turning on a plethora of genes during the very beginning of development is the argument that's being made, like, for that unwinding. I think you would need pioneer factors a little bit downstream. It's like for blindness, you know, neuronal development or eye cell development. You want to get to those little bit more specific ones that unwind and open up. There are more particular groups of genes versus this more broad and generic one. I think it has to be like, this is at the beginning stage, the first step, and then it's just going down further and further in specificity.
Which, in a way, is kind of exciting too, though, because this might be laying the groundwork for more specific studies that have got more, you know, specific implications and stuff.
Yeah.
Is that the feeling you get?
I think so. Like, I think having a developmental map.
In dreams, one is not
Tommy
covered
Plotkin,
by
sleep
earthly
well.
limitations. What does that mean? Come.
Having a developmental map like this and figuring out what genes are opened up and how they're opened up and what's doing the opening up can really help you figure out what it means that a cell is pluripotent. Because if you do have something like that, I can see, like, the next step, right, might be, can I grow a replacement organ? Like, that could be a possible step. Like, you know, you, like, really eventually personalize medicine, because you can figure out, like, from your DNA, Danny, I want to turn on this particular group of things to, you know, reverse engineer from your skin cell a liver. You know, understanding these, like, master regulators could really help push us in the direction where we can do something like that.
Right. That's exciting.
Yeah.
I mean, I know it's far off, but interesting stuff. Like, those implications are... That's some heady stuff right there.
Yeah.
Pretty cool.
Pretty cool. I think it would be very, very exciting to be able to have something like this. It is, you know, baby steps, and it would be a while getting to it, but you gotta walk before you run, right? And so this is one of those beginning things, I think. And it's also fun to think about, like, because this isn't just for human development, this is for any kind of thing, like, you know, how this kind of genetic regulation works in our birds and therefore in our dinosaurs too, and like how those things connect back. Like, it's not an easy thing to think about because, you know, if we don't have access to the genetic information, but it's, you know, it's stepwise going.
Yeah, maybe someday this could help help me to grow a neck that's 5 times longer than my body, so that I could snap up fishes, like in another paper that we might look at tonight.
Yep. And Casey says, do you think healthy organs could be regrown that have been removed? That would be a goal, Casey. Like, not necessarily regrown within the person, because I think in the person you'd still be worried about there being the wrong thing being turned on and off, or like elsewhere in the body. But you can imagine, like, in a dish, taking cells, moving them backward to an earlier state, like this pluripotency, and then putting on the regulators that turn on to create a liver, and then you could implant that, and then you wouldn't have rejection because it's grown from you. So from a personalized medicine,
it's like you're getting an organ donation from yourself from lab-grown cultures in a lab.
Yeah,
that is the dream. People have been talking about that for decades. So
it does. I don't know if you ever saw the movie The Island with Ewan McGregor and Scarlett Johansson.
No, I didn't.
They were trying to do that, but then found out that they were instead cloning people, and that was being utilized for organ research. You wouldn't need to clone an entire person. You would just be in a dish growing up the organ of choice, and then just being, whenever you need it. But in order to grow it up appropriately, you need to know those regulators so you can make sure it's done, correctly.
Okay. Huh. Cool stuff. Thanks for walking us through this. Belinda, when I read this paper, I did not know what was going on, and I feel like the majority of it just sailed directly over my head, so it was really helpful having you kind of guide us through this.
Yeah, I know it's kind of a left field paper, like we haven't been chatting about something like this, and it was very different, but I thought like the concept of the pioneer factor and like what it does in epigenetics is, I thought that was really exciting.
That's cool stuff.
Yeah.
And I didn't know anything about pioneer factors before this, so that's a new introduction for me.
It seems like it's, and maybe this is my naivete, but it's like it was new to me a few years ago. Like, it just so happened that there was a guy in the department who did this.
So it's a fairly niche field even within genetics and epigenetics.
Yeah, just because it's new and it's hard, and for Harissa, like, they don't yet do this for skin grafts. Like, this is still a pipe dream to use for regrowing stuff. But the idea would be one day, like, that's Hanaconda. One reason why we're using pigs too is because they have the regulating factors to grow particular organs, and we don't know what they are, but they will push it to grow in that direction. Versus if we knew what all kinds of factors need to be put on, then you can grow it in a dish as well, like without the external stimuli being needed.
Cool stuff. Who's that you've got there too? I
we have
to detect a cat being
we have Socks here. She's this cat sleeps in the chair next to me most of the day, and then like every hour will come and sit in my lap, get like 5 minutes of pats, and then we'll go back in the chair and just hangs out in the office with me all day.
Sounds like a good life. Yeah.
She she was a stray in like outside of Lita's mom's house, and then we brought her in, and then she's so friendly. Clearly she was let out by someone and just like abandoned, and it's really sad. And then it's whenever I would go over to Lita's mom's, he'll be like, "Where's Belen?" And it's like, "Oh, he's probably like he's been gone for 45 minutes," and I just went and was petting this cat. And so I'm like, when Lita's mom couldn't take care of her anymore, Lita's like, "Oh, we can try to find a home for her," and I was like, "Oh God, we'll take her. I really, really like this cat."
Very lucky cat there. Nice. Now, do you think it's time to move on to the Dinocephalosaurus paper?
Yes, please, if you don't mind.
Let's do it. Yeah.
All right.
Up, in my downloads here.
This was a beautiful paper in terms of figures.
I'm glad you liked it. Yeah, it's a fun one, and I'm not gonna say I have a history with this animal, but it is, It's not the first time I've encountered it, even though it's not a dinosaur. I want to emphasize that right from the beginning.
Mm-hmm.
This is very much not a dinosaur. It lived before the very first dinosaurs. Just a little bit before. But when I was in, I think it was 7th or 8th grade, in science class, we had a thing where every couple weeks we would bring a different, like, science news article to class and talk about it. And the discovery of this animal in the first place, I think back in 2003, this was a news article that I presented to the class and talked about. And so it's nice to see some more coming out about this animal. This is not some critter that was described and then forgotten. We've got new fossil evidence, and it's cool stuff. But I'm wondering, like, what you might be wondering about it, or any questions you might have, Belinda.
Yeah, so I guess the first thing was, as I was reading about it, was that they kept using the word reptile. And I wanted, the first thing right off the bat is, like, I wanted to know if it's correct or not, using that terminology here. Because I know that is a distinguishing feature between, you know, our dinosaurs, our avian dinosaurs versus the ancestral state. So, like, is it used correctly, and, like, how does one know to use this correctly? And, like, where is how do they know? Is it structural that it's more on the reptile side of things? Like, getting, the plesiosaur was coming through my head, right? Of, like, how we talked about plesiosaur.
So it's not a dinosaur, but it is a reptile. But also, dinosaurs are reptiles, and birds are a kind of dinosaur, so birds, strictly speaking, are reptiles too. And I know this will, like, blow a lot of people's minds. Like, this might be kind of confusing or bewildering. But the classification scheme that I think a lot of people learn in school is woefully incomplete. Like, we've got fishes and amphibians and reptiles and mammals and birds. It's like, well, strictly speaking, birds are a kind of dinosaur, which is a kind of reptile, which, if you go back far enough, is a kind of fish. So it's almost like Russian nesting dolls, like, you know, Matryoshka dolls, that you get down to that central one. So this is not a dinosaur, but it is a creature that's related, we think, to critters like crocodiles and dinosaurs.
Mm-hmm.
So, like, it is I think they call it a non-archosauromorph, which is confusing.
Yes.
But that means that it's related to crocodiles and dinosaurs, but it's just outside of the group that includes crocodiles and dinosaurs, which we call archosaurs. So there's archosaurs there, which is crocs and dinosaurs. And then just outside of that, we think there's probably turtles as well.
Mm-hmm.
And these guys are probably somewhere out there. You know, turtles are kind of hovering outside of this group. These guys are probably hovering outside of that group somewhere, too. They're not lizards. They're closer to crocodiles than they are to lizards, but they're not crocodiles and they're not dinosaurs. Does that make sense?
Is this put out by structural similarity to crocs?
Yeah, because we don't have DNA from these guys, so we can't can't look at, genotypic differences. So it's got to be based on morphology. Mm-hmm. So there will be a lot of aspects of, especially the skull anatomy, that show that, like, these are not lizards, and they're not quite crocodiles or crocodile relatives, but they're, like, kind of somewhere in between. And when these guys lived in the Triassic period, Belinda, there were all kinds of crazy, weird reptiles that lived back then, that today they've got no descendants, they've got no close relatives that are still alive today, and so they're kind of a mystery, like, where in the heck do these things belong in the reptile family tree? The Triassic was like the heyday of really weird reptiles that today have no descendants or close relatives. They're just weirdos.
Do we know why they died out?
There was a big extinction event at the end of the Triassic period.
Mm-hmm.
At the Triassic-Jurassic boundary. And that's kind of, dinosaurs show up in, like, the middle Triassic. And then they're kind of small, and the dinosaurs are, they're not major parts of their ecosystem until this extinction event at the end of the Triassic. And suddenly all these other critters go extinct, and dinosaurs seize the opportunity, and they take over the world for the next like 130 million years or whatever. These guys go extinct at the end of the Triassic, along with some other groups of reptiles like that. We still don't know what happened at the end of the Triassic period, but we know that that was the big break for dinosaurs. And potentially a big break for mammals, too. That's like around the time that the first true mammals evolve. But mammals stay in the shadow while dinosaurs really rule the world for the next 100 million years.
It's interesting that for that extinction event, correct me if I'm wrong, but it seems like it was both terrestrial and aquatic.
Yes.
And so it's interesting
both on
to
land
think,
and marine too.
yeah, because like some marine animals have survived for a long time, like our shark descendants, right? But like, it's interesting that this like bevy of reptiles didn't make it.
Yep, it's these guys and the nothosaurs, and I think the thalattosaurs, the placodonts, and the giant ichthyosaurs all go extinct at the end of the Triassic. And we don't know why. Like, maybe there was a great deoxifying of the water, less oxygen. It could be global temperatures rose really fast or fell really fast. Anytime you have like a mass extinction event, that's because there are changes that are happening too fast for organisms to be able to evolve and adapt to that. It's like, it's too much too quickly. Yeah, because, Earth is always changing. Like, it's never static. There's always changes that occur, but sometimes they just happen too fast.
Mm-hmm.
It's like isn't there a phrase about that, about skydiving, where it's like, it's not the fall that kills you, it's the sudden stop at the end. That like abrupt change. Abrupt changes like that occur on Earth are what drive groups into extinction like this.
Yeah.
Yeah.
Yeah, I mean, no, it totally something rapid makes sense. It's just fascinating how much.
Yeah.
And it was non-discriminate in where the animals lived, because like with this paper, it seems like, and I was wondering, like, one of the things I really found fascinating was that they were hypothesized to give birth at sea, like water births. Like, how do we know something like that?
In this particular case, they found an embryo inside. So, like, there's a baby within the abdominal cavity of the adult.
So...
Yeah, and so... Yeah, go ahead.
How... I guess... Again, just my naive take... How do we know it's an embryo?
Because... In this... Like... This has actually happened a few times before with other critters, where it's like, well, how do we know that it was an adult that ate the baby?
Yeah.
Yeah. In this case, it's not dismembered. So, like, and the head is too small to be able to swallow one of these things whole.
Okay, gotcha. So it would have been, if it were intentionally been eaten, it would have been maimed in some way, and presumably bone damage would have occurred.
Taken apart and eaten in pieces.
Yeah. And so
in this case, it's that combined with the fact that it's a marine reptile. So, like, this is a reptile that lived in the sea. And we've got many examples nowadays of different groups of marine reptiles that all evolved viviparity. We know that they also evolved to give birth to live young. We see this in ichthyosaurs, we see it in plesiosaurs, we see it in mosasaurs, we even see it in marine crocodiles. So crocodiles are animals that we normally think of as laying eggs, because all of them today lay eggs. But there were a few lineages of ocean-going crocodiles. Like, take this off the wall. Come on now. This magnet is strong. Hang on. There we go. This is Metriorhynchus. Is a old-fashioned illustration of a crocodile from this crocodilian lineage. It's not like a true crocodile. Like, it's not a member of genus Crocodilus today, but it's a crocodile relative. What you would think of as a crocodile. And these guys evolved to have, these flippers rather than arms and legs. They evolved this like fishy forked tail like that. And we also have evidence that they actually gave birth to live young in the water. So like they are fully committed to life in the sea. And rather than laying their eggs on the beach like sea turtles, these guys would actually just gestate live young in their body and then just like a dolphin or a whale, they give birth to live young in the water, which is
That's
really
really cool.
And this is like essential for becoming fully committed to life in the sea.
Mm-hmm.
Like if you're going to go up and lay your eggs on land, it really kind of restricts you in how truly marine you can be. Like you're not going to be able to develop like fins and flippers instead of limbs. Like you still have to be able to support your body weight on land to be able to produce the next generation. If you don't have to do that, if you can just give birth to live young in the water, you could become fully streamlined, almost fish-like, and really, really take the aquatic thing to its utmost capacity. So,
Almost like lose you're not specialized anymore for land, right? Like any of these adaptations that almost make you versatile for both means that you won't be specialized in one or the other, so you lose one and then you...
It's kind of like that phrase, like, jack of all trades, master of none. Like, these guys can become masters of being oceanic animals. They don't have to have skeletons that support them on land anymore. They don't have to, like, compromise like that. They can just fully commit to life in the sea. And we see this in a bunch of different reptile groups. Each one of these went into the sea of their own accord. Like, these are different events where, like, land-living reptiles went into the oceans and developed aquatic habits and aquatic morphologies. And it's really cool to look at, you know?
Yeah. Do we know anything about parental care with respect to that? Like, the live birth?
That's a great question. Yeah. So in biology, we've got... Do you talk about this kind of thing in genetics, like R strategies versus K strategies for reproduction?
Oh, that's been a while. It's been a while.
Yeah.
Give me a refresher. I remember the terms, but I'm like, oh, what was that?
Yeah. So R strategy, and these are probably something to do with statistics. I don't know what the significance of R versus K is.
Mm-hmm.
It's not radium or potassium. I don't know what it is. But R strategy is like quantity over quality. It's like you just you like squirt out a bunch of young and they're all really small and underdeveloped. You don't exert a lot of parental care or anything, and you just, you know, think of it like kind of like a dandelion, where it's like it's got all those little spores with the little fluffy things and they just fly off in the wind, and you hope that like one of them will land in a spot where it'll eventually grow up to be a dandelion, but like chances are none of them will. It's like why dandelions produce a lot of them like this.
It's like you overwhelm the system with offspring so that if a few make it, then that's enough to have passed on your genetics.
Exactly. Yeah, and it's
Cicadas.
like you don't invest a lot in each one. They're cheap and you do a lot of them.
Yeah.
As opposed to like
cicadas. Yeah. Cicadas, for example, right? Like they have no defense against predators, so the way you overwhelm is you just unload everything, and eventually a few will mate and pass on.
Yeah. And the opposite end of that is like what we call a K strategy, and a classic example is like an elephant, where it's like an elephant, they get pregnant with one offspring at a time, and that might take like 18 months to gestate, or 24 months, whatever it is for an elephant. It's a crazy long amount of time. And then after that one offspring is born, it stays with its mother and drinks milk, and like there's a lot of parental care invested in it.
Mm-hmm.
So it's like it's quality over quantity. It's like you really pay a lot of care to this one offspring, and you're banking on that paying off for you to get your genes into the next generation. So that's K strategy. Sea turtles today, you can think of them more like R strategy, where they just have a lot of babies and like a lot of them don't make it. In fact, the vast majority of them don't make it. It always breaks my heart. I love sea turtles. Baby sea turtles are so cute, but most of them don't make it. You know, it's interesting in that like sea turtles today are marine reptiles. They're reptiles that live in the sea, but they're different from these guys, like Dinocephalosaurus and Mosasaurs and Plesiosaurs and Ichthyosaurs. They gave birth to live young, which means that you've got one big offspring gestated inside you, and then that implies parental care. It implies that offspring is probably staying with the mother, maybe staying with a family group for a long time afterward and receiving a lot of care. So we don't have direct evidence of parental care here, but it's part of that kind of paradigm where, like, it makes sense for them to have had more parental care because that's the pattern that we see in animals that have one offspring at a time. Does that make sense?
Yeah, it's really cool to think about, like, that you're getting that resolution of insight on something like behavior, right, and care just from, like, a skeletal structure.
It's inference, so, like, we're still guessing to a certain extent, but it's like it would be really wild if these guys didn't have any parental care. That would be totally bizarre based on all the other critters that we see that have the same kind of reproductive strategy, you know?
I get... Where is your take on this? Like, the long reproductive time could be a way of parental care as well, even if it swims away as soon as it's born, right? Because
and there's a lot more development that's happening in the womb like that. Yeah, so you could almost think of that as parental care, I guess.
I guess it's like, have you heard of it, like, in humans, there's the 4th trimester, right? So, like...
No.
So, in developmental biology in humans, after a baby's born, there's like, they don't see well when they're born. Like, everything's blurry for months, or...
They don't do anything particularly well when they're born.
No, well I mean, for years, too, right? And possibly even after 30 years, you know, not doing something well.
But I feel a little called out, Bullet. My goodness.
Ask my father, you know? But, like, the development of the embryo can't finish developing in utero. There's just not enough space. Right, and so that's why there's this 4th trimester now, which is the finalization of that final, like, 3 months, where afterwards, like, the vision of the baby is that, like, they still can't take care of themselves, but that's, like, there's at least, like, a little bit more time we've evolved to the point where we can't finish developing internally. And so I wonder, you know, like, extra parental care, if you let the animal continue to develop and you're protecting them inside the womb, like, it almost feels like a way of, like, it is direct parental care, but it's indirect to the organism. Like, to the offspring, in a way. So it's like, I think even if they did swim off right away, there's still been a lot invested into the animal, to that offspring. And so it's still going on that quality, how you're saying. And that actually also implies that their lifespan is probably pretty sizable, right? Because, like, versus the really small mammals that just laid a bunch, or, you know, like you know, have a huge litter and then some survive and some don't and you move on, versus, like, these, like, who knows how long the gestation time was, but that signifies that the animal had to have lived a significant amount of time.
Yeah, because if you're only going to have one offspring per year or something, or maybe even fewer, chances are, like, you need to have a long life in order to ensure that, like, you can achieve replacement rate for your population. So yeah, it does imply a longer lifespan as well.
Yeah. Azure Fire asks if there's something similar to mouse development to human. Yeah, so Azure, it's like there's additional time that mammals need to finish developing that's now become outside the womb. And that's, you know, it's also an argument of, like, the size limitation, just like the evolutionary pressures that, like, select for more intelligence or select for these additional behavioral complexities. Like, it's just too much to finish taking place in the womb, where, because the organism, it's not, you can just focus on the brain, right? The organism just keeps growing, and eventually you can't, like, it's not that there's not enough room in the womb, it's you can't give birth to something. Like, eventually a physical barrier does happen. Like, you can no longer give birth to that size organism.
The birth canal is restrictive like that. You can't have too big a head or it'll get stuck, you know?
Yeah. And so, like, it is, like, there is this developmental psychology argument of, if you were to start doing C-sections on everyone, would you eventually get to the point where they just won't trigger, you know, like, labor because you're just taking people out, right? And that way you can develop longer. That's a, you know, a long, far-fetched hypothesis, but that's, who knows if that's something like that would happen.
And there's an interesting thing that happens with other mammals, like marsupials, where have you ever seen, like, a baby, like a newborn kangaroo, Bullet?
The really tiny jelly bean looking
Exactly. Yeah, and so it's like they're born as basically like a larva or
Yeah.
like pre-larval, and then they have to like they've got these tiny little rudimentary arms and they're able to use those to crawl up into the pouch, and then they actually finish their or they continue their development there in the pouch after being born, and yeah, it's interesting. Like in different groups of vertebrate animals, you have different strategies like that, different degrees of parental care, and it's just interesting, like all these different strategies that have evolved, that all work in their own unique ways.
Yeah.
And so it's cool to think about these guys like as reptiles, as sauropsids, rather than mammals, synapsids, and like the different strategies that evolved in Sauropsida within reptiles over the past, few 100 million years. And like very few of those are still around. Like we're almost kind of impoverished nowadays in like the number of reptiles that are still around and the strategies that they have. They all seem pretty homogenous. It's like, no, they all lay eggs, except for some lizards and snakes that give live birth, but they're pretty rare.
So it's almost like the birds, right? Like we're almost pigeonholed into the kinds of birds that we have because of the big extinction events, and so it's like the diversity that you would,
this bottleneck like that,
yeah. It's kind of frustrating, right? It'd be kind of cool to see some some further diversity, or like I guess that's one of the cool parts about like how you said studying like the Antarctic, right? Where it's like the undiscovered, like maybe we'll find some new things there too, and I wonder like what reptiles could be in that area as well. One of my favorite part was like in this paper that there were 7 different fossils that they found.
I know,
of these and it was like, whoa, that was cool and like not expected.
It's some really neat stuff. Yeah, and hopefully it'll help us determine where these animals kind of fit in on the family tree as well, because like this looks very similar to another creature called Tanystropheus, which it's also got a ridiculously long neck like this, but it's got way fewer neck vertebrae, and so this guy also, it's also got an absurdly long neck, but it almost seems more flexible than the neck of Tanystropheus, until if you look at this image that you've got behind you, Balint, you look at those cervical ribs right there. So these like long, thin rods that come off of the neck vertebrae.
Is it? It's these.
Yeah, those are what we call cervical ribs. So I think developmentally those are actually tendons, or maybe ligaments. I'm not totally sure. It's one of the two, but they actually turn to bone as the animal grows and matures. They're almost like ossified tendons, and so those act as stiffening agents in the neck. So like, if you have long cervical ribs like this, it means you've evolved to have a really long, very stiff, straight neck. It would limit the amount of flexibility in the neck there. And so like the pose that you see right here, that's after the animal died and its flesh rotted off and like it can kind of curl up. It would not be able to do this in life. That's like, it did not have that degree of flexibility. Because all of those would have to be, like the stiffening rods that you see there would have to be inside the skin.
Is this something similar we talked about like last week or 2 weeks ago about like a way to age samples is the presence of these calcified tendons, right?
This would probably be a mature animal, or fairly mature at least, in that like a baby or a juvenile might not have turned to bone yet if the animal's not mature. Yeah.
So I wonder if there is some meaning behind that in terms of benefited flexibility in the young animal versus this more like strengthened in an older one. Like, could prey be something where you need a strong neck, otherwise your neck breaks?
I think a large part of it is just like, as an embryo, you got to be curled up, so like you have to have a more flexible neck.
Oh, okay.
Yeah, and then later on, when you're developing into an adult and you have to catch food on your own and stuff, because the authors aren't really sure in this paper, and I found this really charming actually. They're like, we don't know why they have these ridiculous necks. Nobody knows yet, but it probably has something to do with feeding, given that that's like the business end of the animal. I like
that they said they don't know. Well, Let me let Socks out. He wants to.
Yeah.
He's been having.
That kind of like, candid admission there is refreshing in a paper like this, and I appreciate it.
It's. I mean, it's
really...
nice. Like it's hard to ID some of these things, right? And so having that level of like self-awareness and admitting that like we just don't know, but here are maybe some hypotheses is really cool.
Yeah. And it's interesting that in like other marine reptiles also sometimes grow these ridiculously long necks like this. This happens multiple times within different lineages of marine reptiles, and we don't really know why yet. Like we've got these fantastically, stupidly long necks in these critters, and it's some kind of a mystery. Why?
Yeah, it's a beautiful fossil.
And I find that really exciting.
It's.
Yeah.
This is what you want, Lita? It's a beautiful fossil. Like
gorgeous.
I was blown away by how, because it looks a lot like that juvenile T-Rex that we saw, almost in terms of the quality of the fossil, where and you saw what's inside of its like digested, right? That we were looking for like the different niches of where they feed over time within age, which was I keep talking every week. I bring up that paper because of how cool I thought that it was, right?
I'm so glad. It's
just
really
like
favored.
from like quality wise of like a fossil, it seems like it's almost
they
like
lived tens of millions of years ago, or hundreds of millions of years ago. In this case, this is like 240 million years old, and yet it looks like that.
Yeah.
I mean, that's extraordinary.
The other thing you were saying, like with that other species in terms of its similarity that it's like very close to, that I thought was cool, was that that other similar species, unless I'm misread the paper, lived at a very similar time, right? And like was very like you said, it had a super similar, except with like these structures, but like size wise, it was also similar. And not only the same timeline and same size, but same geography.
Yeah.
And so that gets like this forever question of what is a species, right? I wanted your take on how sure would you suggest we are that these two animals are two different species?
Oh, they're very different. They're very, very different. They've got different shaped heads, different bones, like arrangement of different elements in the head, different numbers of neck vertebrae. Like they might not even be in the same family or superfamily or same order or something.
Okay,
like
so
that.
like pretty just
be...
from the morphology, we're like, it's not just like slight
animals.
Differences that made them.
It's not like they had a super recent common ancestor. Like these are critters that are separated by who knows how much time. A lot of that stuff is still a mystery, but what it does tell us is like there's something going on in these Triassic ecosystems where like it really behooves you to grow a stupid long neck like this with a little head and try and go after fishes probably. It does kind of get into it's like I don't know, it would be cool to see a paper about this at some point, but like how the closer you are, the more likely it is that you will have convergent evolution that brings you to a similar morphology. Where it's like, an animal like fish is probably not going to develop a super long neck like this. But there's something about this group of like archosauromorph reptiles where even if you have, two that aren't particularly closely related at all, they might share a common ancestor like tens of millions of years ago, they might still develop very similar morphologies because they're still dealing with that basic starting point. They've both got this kind of like almost lizardy kind of morphology to start with, and so they've got the same starting point for like evolution to kind of shape them in similar directions. It's like convergent evolution can be really powerful. Like you can get the same morphologies that show up from disparate ancestors, but like the closer those ancestors are to each other, the more similar the endpoints will be if they're converging toward one another.
Right.
Does that make sense?
Yeah, and that's why you
Yeah.
have, like you said, something's happening with some kind of benefit of this long neck, and is
Yeah.
it then weird that we don't really have aquatic animals like this? Like in chat mentioned like the possibility of like maybe like a heron, right, with the long neck that's stable, right? But
Yeah.
I can't think of an aquatic animal that really resembles this. And so is
Really
it?
don't have anything. So a heron has got an extremely flexible neck. Like herons can practically tie their neck into a knot. It's
Okay.
it's ridiculous how flexible the neck of a heron is. These guys, their necks were not flexible, and like this animal is dead, and this is not a natural pose for it to be in. Like it would have a very long, very very stiff neck, and we really don't have anything like that today in the oceans.
Mhm.
Something with just this really long, ridiculous neck that's very, very stiff. It's like you're swinging around a bunch of broom handles that are, tied together or something like that.
I like that.
Yeah, it's like we have got no good analogy for this today. No animals like this today no animals today exhibit a similar kind of strategy to this animal, and so that's why it's kind of a mystery. We still don't know what exactly that long neck was for.
And it's interesting we keep seeing it, like you were saying. There keep being more and more instances. And then
we get this in plesiosaurs, your plesiosaurus, Balint,
Yeah.
and in Elasmosaurus, like they have a lot more neck vertebrae, but their necks also would have been very stiff. Their neck vertebrae are almost this kind of like double barrel shape, almost like if you were to take a double barrel shotgun and like chop it up into a bunch of pieces, and then so it's got like these double barrels that like you put them one after another, and it's hard to really get them to like flex because they're double barreled like that. That's kind of like what plesiosaur necks are like, with the long neck plesiosaurs, especially the Elasmosaurs. Their necks were really long, but they're also really stiff. It's like, what is that for? No animals today have something like that. It's bizarre and unique, and it's a mystery.
Do you think that their prey may have also gone extinct? Because I wonder if it was like a hunting strategy almost, and because I guess it reminds me of the hell ant, right? Like there was this vertical mandible that nowhere else around today do you again see. And it was hypothesized they got so specialized for their prey, and when their prey went extinct, they went extinct.
Yeah.
That might be a totally naive way of thinking it, but it's interesting of like, you're seeing it so often. There must be a reason for it. And it's just weird that it's gone. Like, why hasn't it like shown up again?
Yeah. Generally, when we're thinking about animals like Elasmosaurs or Tanystrophiids or Dinocephalosaurus, like this guy, we're used to thinking of them going after fishes and eating them. Maybe squid too, maybe. But fishes are usually like that's what we're thinking about. Just kind of smallish fishes. But you always have small fishes. It's not like small fishes ever just blink out of existence. They're always there. So it's probably not prey specialization. This could be a strategy for catching prey that we just don't see in animals today. There's an idea that goes back a long time that like maybe these animals were kind of sneaking up on schools of fish, and the smaller you are in profile, the less likely it is that you'll scare fish away. So like one little fish swimming up to a school of fish is not going to scatter them, but if you if you sail a cruise ship up toward them, like they're going to get scared. So like the smaller you can be in cross section, the more likely it is you can just kind of sneak in there and grab them. And so the idea is maybe that like these guys can keep their big bulky body way far behind by putting at the end of this really long neck, and they could just kind of like sneak their head in there and grab one, and then sneak their head in there and grab one like that, because it's so long and so narrow that like they can't even see the body way back there.
And I mean
that's
that
the idea. And
that again leads to like hyper specialization, right? Is like the
potentially
the imagery that I'm seeing. Like it's a really cool feeding behavior, but it's almost like you're a particular kind of prey is best suited for that, right?
Maybe, yeah. And also, like, how in the world do you test that? Like, do you build different kinds of robots and then, like, with really long necks, and then, like, see if schools of sardines will be spooked by a certain kind but not by another kind, or like it was really tricky, you know.
I think that's too hard, right? Because if I could always, if they're not spooked by any of it, couldn't the argument be made that the prey had just died off and you're getting at something that doesn't have the innate fear anymore because it's not there?
Sure.
So I guess it's similar, but maybe that's how you would, right? Like, I'm just thinking of ecology papers where, like there's the caterpillars that have those distinctive markings on their back that look like eyes.
Like eye spots and stuff?
Yeah, and it's like the question is, well, what is the function of that when it comes to a predator? So what they do is they put out Play-Doh that's modified in a way to either be just solid green or with like that eye spot, and then they just see how many birds pecked at it. And there's a lot wrong with the experiment because you could argue it's like Play-Doh and it's like the smells and the movements and blah blah blah, but it does seem to be like the birds peck the green ones far more than the ones that have the eye spot, even though it's like they're all fake. So it's a really interesting question.
Yeah, and I don't know. I find this sort of thing really exciting because it's like we still have mysteries all around at this point. And like it feels like this is something that's ripe for somebody to figure it out, and it hasn't happened yet. Like, I'm just waiting for the day that somebody deciphers this and comes up with an idea that, in fact, actually, in I think 2003 when this animal was first described, there was this idea that like maybe the long neck also helped with suction feeding, where it's like if you're maneuvering your head up to a school of fish at the end of this super long tube-like neck, you can just kind of like open your jaws and go, oh, and take in some water.
Okay. And
then you're like pulling a fish in towards you, and that really, really long neck actually helps with that kind of like suction.
That's cool.
Backward, because it creates a longer vacuum there.
Yeah.
And that could be part of it too. Like, yeah.
That's a brilliant hypothesis. That would be super cool if that's what it was.
And how do you test that though? That would probably be easier to test than the other one based on like fish behavior.
Yeah, I mean, you could build and demonstrate probably the vacuum and the amount that it would go, but then then the question becomes, well, what do you do with the excess water in the animal? How do you get it from having osmotic shock, because that's a possibility where the salt balance is off because too much water into it's hard. It just, I think, illustrates how difficult this science is, but also how amazing that we're this far, that we can be making these kinds of, this is what we're hypothesizing about, like not what the animal looked like or what's related to, or even how it lays its embryos and its possible, like we're going like really deep into its behavior, and it just shows like how important I think this find is. Yeah, it's exciting stuff, and it was cool that this got so much media attention as well. We were talking about this the other day on my stream, how I was a little annoyed at how one of the authors in a press release, they said, "Oh, it's very dragon-like" or something. Yeah. And sometimes, oh man, the news media gets a hold of a word like that, and they just blow it way out of proportion. And so, oh, dragon this and dragon that. Scientists found a dragon fossil, dragon dinosaur, blah blah blah, and it's just, it could be a little bit maddening, especially when this stuff gets spread across the internet, and you get people who are, I don't know, a lot of people who want to talk about leprechauns and unicorns and giants, and then then now they've got dragons to play with, and it's just. Yeah. There are definitely downsides. You might get a lot more headlines if you use the word dragon in your press release, but there's definitely the potential for a lot of miscommunication and people taking it in weird directions and stuff like that. So it's like it's a word that captures the imagination, which can be a good thing, I think, when for not from a media perspective, right? Like if you're hearing from a scientist, like it's a really cool illustration and could get someone excited about the topic. But I think it's like you said, when it's the media grabbing hold of it, it can be twisted in a way where you're reading something very different than the original intention of the comment.
I got a text message yesterday during stream. Was it yesterday? It was yesterday. From my mom, or the day before. It's on Monday. She's like, "Oh, Danny, did you hear about this? They found a dragon fossil." I'm like, what? My mom heard about this, and she's texting me about it and wanting to know about it. And, yeah, I don't know. It's a mixed bag, I suppose.
Yeah.
You know, it's a double-edged sword. Like most good swords, very few good swords have on one edge, right? Maybe a samurai sword.
Well, I mean, Darth Maul would tell us that the double-bladed saber is the best thing, you know. When that came into theaters and I saw this guy come out with double-bladed, I was like, "Oh my, what is this?" George Lucas blew my mind.
Isn't that a four-sided? Because it's got two sides per blade. So when you've got two blades, that's four-sided, isn't it?
I mean, he blew my mind again when the robot General Grievous, like in the third movie, split his arms and then he had 4 lightsabers individually. I
also thought it was really cool. Yeah.
It's flashing laser swords. I'm great.
Yep. Anyway.
I had one other big question about this paper, and just I was wondering about the area where it was discovered, because they discovered the other species here, right? That was like a close relative. They discovered 7 of these fossils. Is there something special about this part of Asia that is either understudied or just some kind of hotbed for a particular kind of ecosystem, or was this just kind of like a luck of the draw?
I think it is kind of a luck of the draw thing, and China is a big country, and it's got a lot of exposed sedimentary rock in certain areas, and so it really is like a luck of the draw thing. Yeah. Tanis Tropheus, I think, originally was found in Europe. I want to say Switzerland or Germany. I don't really remember. But yeah, these critters would have lived all over the world, presumably. It's just you got to find rocks of the early Triassic in order to be able to find these critters, and those happen to be exposed in certain parts of Europe and certain parts of China. So it really is kind of a luck of the draw kind of thing. But these are exquisitely well preserved, and part of it is a luck of the draw thing, but also part of it is that paleontology is well funded in China. You've got a lot of up and coming paleontologists there, and that country's really pushing for the development of this science, and we're going to see more and more and more of this sort of thing coming out of China in the near future.
Is that coming from a sense of almost how we talked about this national ownership of this is our science, is what kind of we're representing, and we kind of want to be...
Yeah.
It's almost a sense of national pride in terms of what you dig up.
I'll preface this by saying that I have never been to China. I do not speak Mandarin or Cantonese. But my sense as an American, reading these papers and talking to people who have been to China, who are doing research there, is that China is really, really pushing to modernize, and they are pushing to really excel in the sciences. And people who are in charge in China realize that dinosaurs and other fossil critters spark the imaginations of young people. And so they're trying to build a new generation of scientists by investing in fossil science right now, because they recognize that the more fossil discoveries you make, the more you get young people interested in science, and the more future scientists you create through that by getting kids interested in science from an early age. So it seems like, from where I'm standing, it seems like that's what's going on there, and it seems like it's working.
I mean, that's brilliant. That's what you're doing here on Twitch, right? Not in a national sense. I mean, you're spanning international waters, Danny. You're a global icon at this point, sir. But yeah, it's the same idea. I think dinosaurs are a great gateway to other sciences. I feel like a lot of us started off really interested in dinosaurs and paleontology, and it's just either
dinosaurs are a gateway drug into the sciences. Absolutely.
I mean, accessibility. I wish I had accessibility to you as a kid.
know.
Because right there, I mean, right, like there was no asking, like when are you going to ask a paleontologist questions, right? And so it's, I imagine they're investing then in museums as well, and like accessibility on a very broad.
It's crazy the number of fossil museums that they're building in China. It's really astonishing. Yeah, makes me envious as an American paleontologist.
Yeah.
Oh, yeah.
Yeah, but no, this was a super, super cool find. Like, I really liked this paper. I was, again, I had a couple of naive questions about it, but I was just, again, blown away by how cool the fossils were and how well intact they were, and like what all we can learn about them, and again, the fact that there were 7.
Yeah.
Silly thing to be impressed about, but just how we've talked about, like, honestly, one really good fossil, like you've said, is a great way to identify a species. But to have 7?
It's pretty awesome.
Yeah.
For sure. And if they have 7 that are here in this paper, that probably means that they have 4 or 5 times as many that just haven't been prepped yet or haven't been properly studied. Like, this means that they will have more of those just kind of waiting in the wings.
Is that really for, like, the follow-up papers that you've mentioned where there's... You've said it's like a commonplace thing where there'll be like this. I think there was one that we talked about last week where there was a really big paper on one species that came out, and then we never saw the next follow-up to it.
Yeah, it happens a lot.
Yeah.
So it's nice that, again, it took, 21 years, I guess? But 2003, this Dinocephalosaurus, this genus of really cool non-dinosaur Archosauromorph, was first published, and now we've got a follow-up with so much more information, and we will probably have more in the future, and that's really cool. So I don't know, it's nice to see this kind of come to fruition in that sense.
Yeah.
Like, I've been reading about this critter since I was a kid, and and now we've got more of it, and that's really exciting.
Still lots of unanswered questions, like you were saying, with like the neck and the feeding behavior and just a bunch of stuff we came up with.
Yep.
Just like chatting between each other, but it's super, super cool. So
Isn't that
really,
neat? What a weird time too. The Triassic is just. I know people who work in the Triassic, and they're almost like evangelistic about it. They're like, the Triassic is the coolest, weirdest time. It's so short, but you've got all of these bizarre reptiles that evolve, and then they're just gone. And like, everything in the Triassic is super bizarre and super interesting, and then poof, vanishes at the end of the Triassic, and then dinosaurs take center stage and hold on to it until the end of the Cretaceous, like 130 million years later. And that's, there will be a lot of cool papers that come out about Triassic animals, I think, in the next few decades, and I'm here for it.
Nice. I hope we figure out why that extinction event happened too.
Yeah, it's still a big mystery. We still don't really know. It doesn't seem to have been an asteroid, probably. It might have had something to do with global warming or global cooling. But out of all of the big extinction events, the Triassic might still be the most mysterious. Because it is one of the big 5. The more we learn about these things, the more we learn about how we could potentially avoid extinction for ourselves and the rest of, our neighbors on this planet. So it's very, very important to understand these things for our own well-being and continued existence on planet Earth. So
I think that goes unseen of the implications that we get from studying these, right? Because I feel like maybe that's a short-sightedness of, this is a dinosaur that's been dead for a long time, why do I care? And like you're saying, if an entire ecosystem just went out, and we don't know why,
globally, biomes around the entire globe, you had more than 50% of all species on Earth died out suddenly. Like, understanding that mechanism there could be the key to saving our own skins at some point. So like, it's way more important than I think most people realize. So like, we need more work done on critters like this. It might seem like it's just cool for coolness sake, but it could really be instrumental in preventing our own extinction sometime.
No, it's very. I like that summary of it. It's just kind of, again, why funding for these things is important.
Yeah. Should we leave it there, you think?
It is 9:30 for you, Danny.
Oh boy.
Which I'm so sorry.
12:30
We
for
just
you. Holy cow! It's already tomorrow where you're at, Belen. Holy moly.
I get wrapped up and I have so much fun chatting that it's just I lose track of time.
Yeah.
But Danny, I really, really appreciate it. For folks who are here next
Likewise.
week, we're have to skip next week because I'm off at a conference in DC. But we'll be
Nice.
back the following week.
Presenting before the Pentagon, or
No. It's our big once a year conference. Well, usually it's multiple organisms conference in a year, and this time everything's pushed into one conference. And so it's like this big, virtually starting tomorrow and then in person on next Monday through the Sunday. We're actually meeting on Capitol Hill with politicians on Tuesday to advocate for science funding.
Good luck with that. You take it to him, Belen. Holy cow! Let's get some science funding.
Yeah, the cynic in me is like, oh.
Who knows? We need it.
Yeah. Oh no, definitely. But we're having that on Tuesday, and then the conference events through the week, and then we'll be streaming on some of the days next week. Lito will be running it from here, and then I'll be back on Sunday, and then so next week, y'all. Unfortunately, no crossover. Even though my highlight of my Wednesday is gone. But then
we've always got the Wednesday after.
Yeah. But yeah, we'll be back the following week. Danny, thank you so much for coming on. It's always a weekly highlight for your time, your insight, for selecting these papers and just on it everything you do, my friend. Thank you so much. And if y'all are not following Danny of Paleontologizing, please go drop him a follow right now. Easiest follow you'll ever do on Twitch.
Appreciate that.
Thank you, Danny. If you only follow one person, chat, on Twitch, go follow Danny.
Well, hopefully that means they're already following you, Belen. Thank you again, and good luck at the conference. Seriously, advocate for some science. I'm excited to hear about it.
Yeah, we're gonna be. It'll be interesting to see what we come out of it. I've done virtual advocacy before, and it's a very mixed bag. Sometimes the administrator, the politician, doesn't even turn on their Zoom camera. I guess if it's in person, they have to, right? But it'll be interesting.
There you go. Yeah, well, hopefully some of the skills and enthusiasm that you've honed here on Twitch will shine through in person like that.
Give them some stickers that'll get them hooked, right?
There you go.
Danny, before you go, if there's anything, what's coming up on Paleontologizing that people should be excited about?
Yeah, we've got Thursday Birds Day tomorrow, as we always do on Thursdays, and maybe Friday, Dinosaur Fossil Hunter, that paleontologist simulator game. They've got a new level. They're releasing some new content, and I'm going to be playing that soon. I've been talking with them, and so we'll be doing that sometime soon as well. Probably Friday.
Nice.
But we'll see.
I do enjoy. I love the video game dissection streams that you've got, Saurian and
It's been a year or two since I've done the last one, but we'll be doing that again soon.
Nice.
Maybe Friday.
Awesome.
Yeah.
Thank you so much, Danny. I'll see you tomorrow, my friend.
Thank you, Belen. Take care.
You too.
See you around.
Bye, Danny.